Thank you very much, David, for the very kind introduction. Thanks to the entire organizing team and also to you for being here. I would like to take you a little bit into the area of trait prediction from genomic, metagenomic data. That's what I was asked by the CAMDA organization team, and I would like to mention that this not only relates actually to the antimicrobial resistance challenge, but also, for instance, to the microbiome health challenge and maybe also future challenges. Feel free to interrupt me at any time with questions, so we can keep this very open from my point at least. And of course, I'm also very open to questions in the end. As David mentioned, I'm a chemist by training, so I'm mainly looking at the kind of biochemical and biological problem. I don't care so much about the particular solution as long as the solution is good and helpful. And I think that's what is a good starting point also into challenges like this and assessment projects like this. So here, my starting point are, of course, microbial communities. We all know what the importance actually of microbial communities is, ranging from across all ecosystems on Earth, from very common like host-related ecosystems like us, plants, soils, ranging to even very extreme habitats. Interestingly, the cooperation of microbes is very different in these different ecosystems, but almost everywhere, except some very, very extreme environments, we find multiple microbes filling different roles or having different traits actually that allow the microbes to survive there and to just settle in these environments. And for this reason, as a disclaimer, I'm not looking at phenotype, trait, role from a very kind of conceptual point of view. For me, these are kind of expressions of features that we would like to understand and we would like to study to learn how microbes can, as a whole microbiome or also alone as a particular species or culture, survive and proliferate in this particular situation. Sometimes this is more closer to a trait, sometimes this is more closer to a phenotype. So please don't nail me down actually on these terms. The baseline actually for studying microbes, you all know, or most of you at least know, is actually cultivation. So most of the knowledge actually that we have about microbes comes from cultivation-based studies. And in cultivation, we typically pick cultures or co-cultures, depending, for molecular analysis, for molecular sequencing, for instance, genome sequencing by particular interesting traits. Obviously, actually, antimicrobial resistance, for instance, would be sometimes a trigger. If it's an interesting type of resistance, then we would say, okay, let's sequence these genomes. So this was the way, and is still the way, how genome sequencing is done. So often actually just in surveillance or in medical situations, we pick certain microbes for sequencing. Metagenomics, so the study of microbial biomass without cultivation, without separation of the species, turns that question kind of around and provides us a bioinformatic problem. Because we are able in metagenomics, as you all know, we are able to sequence the DNA from an environment. This is not fully unbiased. We always know that the kind of DNA extraction protocols breaking up the cells don't work in an unbiased way for all members of a microbiome. I will not touch this in detail, but in principle, these techniques are very established. And with bioinformatic approaches, I will a little bit at least get closer to them. We are able to reconstruct partial genomes. They are almost always partial, so very rarely we can really get a complete genome out of metagenomics, but typically these are partial genomes of a reasonable quality that have sufficient quality to study them. And in this case, we just know the microbiome, or we know the sample, or we know kind of the biomass that we have used, but we don't know each of the members of these microbial communities. We don't know their roles, their phenotypes, their traits, as said before. So what are they doing, and how are they collaborating, and how do they settle in these environments. Just to mention that metagenomics is not the only way. Of course, in particular in medical settings, we also have kind of techniques in between. So for instance, like targeted genomics, where we particularly target certain cells by cell envelope features, by molecular markers or so. So it's possible, of course, to kind of enrich from, for instance, from a patient sample, very few microbes of a certain type that we're interested in, and then also to sequence them. And here, the particular question, for instance, of risk assessment could be asked, which is kind of also related to the questions before that I mentioned. So we would like to be able in the next years to already predict, based on molecular findings, the potential risk, for instance, of a bacterium or of a virus, for kind of creating wider problems or bigger problems up to a pandemic. Nobody can predict this reasonably at the moment, but we would like to at least be able to go better in this direction. I will not go into method details, as I mentioned, but I would like to mention that along this way, actually, from learning how metagenomics can be done, and metagenomics is done computationally, along with this, there were assessments, so we'll come to that later, and there were standardizations. And for me, actually, as more kind of a user of methods, or as a person who's more interested actually in the biological and biochemical aspects of them, that goes together. And which would, of course, also be kind of a request, or at least a wish from my side to the CAMDA community, to not only engage in assessment of methods, but also engage in standardization. We really all benefit from standards. And in this area, I just mentioned that the standards for single-cell assembled genomes, the MiSeqs, and the metagenomic or metagenome sample genomes, the MiMax, they were extremely helpful for sharing data. Without these standards, it would never have been accepted by the big DNA databases, for instance, that people upload metagenomic bins to these databases. And so here, along with the assessment, actually standardization was very helpful. The assessment here was initiated when we started to be able to do reasonable large-scale metagenomics, and the assessment here was the critical assessment of metagenome interpretation, CAMI. It had two experiments, CAMI1 and CAMI2 so far, and these were very helpful to explain us and to show us actually how far the computational methods are able to sort out the different bacterial genotypes actually out of a metagenome. We have learned a lot of kind of limitations that were to be expected. That was entirely clear, but we learned that these limitations could also at least partially be overcome by using the right computational methods. This is just a one-slide summary of CAMI1. And CAMI1 actually confirmed a process that had started already in 2012, 2013, that large-scale metagenomics is possible. And this will be the platform for the trait prediction that I will show you later. And as an example for the standardization, you see here, again, important as input for trait prediction, these are different quality classes of partial bacterial genomes from finished. This is typically not getting out what we get out of a metagenome. High quality drafts, this is what we mainly would like to work on. Medium quality draft can still be kind of used for at least partially, and low quality draft, they typically cannot be reasonably used, also not for trait and role prediction. They are defined clearly by completeness and contamination percentages that shows an estimate actually of the purity of a metagenome, of a bacterial genome or microbial genome reconstructed from a metagenome. So with that, I hope it's kind of clear that the question is important, that the question can also be worked on. Metagenomic data are currently available, definitely on large scale. So my group has started to work on this problem actually in 2012. So when it was clear that we were changing from the kind of initial years of gene-centric metagenomics to the genome-centric metagenomics. Immediately when this turned out to happen, students and colleagues in my team over years actually started also to work on the interpretation of these metagenomic data. And that's what I would like to introduce you now in a little bit more detail. The trait prediction is, of course, nothing that we had to invent. So trait prediction is a classical kind of task in the annotation of microbial genomes and has even been thought about before, as long as it's linked to marker genes. You know markers very well from antimicrobial resistance, but there are also many other traits, mainly metabolic traits, that are linked to very few genes that have the key molecular roles actually in these traits. I gave you here one example actually, which is the ammonia monooxygenase, which is the key enzyme that is responsible for anaerobic ammonia oxidation, and so these we find this marker gene everywhere where ammonia oxidation can be performed by a microbe. This is, of course, not the only gene, but it's the central gene, the core gene, and it's typically flanked by a couple of other enzymes, regulators, and so on. But we can use it as a marker, and this has been used, of course, since many years for this particular trait. For instance, PCR assays have been established, so we can use amplicon studies. 6-8S amplicon studies certainly, but also the AOA, ammonia oxygenase, the MO gene, has been used for amplicon, for function gene amplicons, and so we could screen actually microbial ecosystems for potential ammonia oxidizers. That's working very well, but unfortunately, many other traits don't rely on these kind of single core genes that we can just use as markers. And so it was clear when we were thinking about trait prediction of microbial metagenomic assemblies that we might need an alternative. I just summarized a little bit the rationale behind. As said, we started when genome-centric bioinformatics and genome-centric metagenomics, became feasible. From that time and not earlier, the metagenomic bins were representing nearly complete genomes. Marker genes, even if existing, or even if they are present for a particular or central for a particular trait, they can be absent from a MAG. That's important to know, and sometimes this can even be a systematic kind of effect depending on the strain diversity in the metagenome. So sometimes we can even specifically miss a marker gene, but all other flanking genes might be there and might inform us about the trait. But just to rely on single markers is dangerous actually for partial assemblies as they come out of metagenomics. But as said, also many traits lack simple markers. I gave you an example here on the right side. This is a bacterial secretion systems, and we know that bacterial secretion systems, they're central for the interaction of microbes and hosts, but they're quite flexible. They establish as large protein complexes, and these complexes are kind of flexible in terms of their building blocks and their units. So sometimes you see even here, these are cluster of orthologous group numbers. You see that there are alternatives listed for certain subunits actually of these complexes. And this makes it very difficult to just link the presence at least of a functional secretion system to a single gene. This is not a marker gene anymore. It's a big catalog or a big matrix that can be sparse. But of course, the genome as such, the genome completely is a major source of information. Even without knowing the actual transcription, often the discuss about the kind of value of a genome out of a metagenome and its information content, but we know, fortunately, actually, that bacteria quickly lose information, and this information that's lost doesn't show up anymore and doesn't dilute the information too much. As we know that the scale-up of metagenomics allows us to sequence metagenomes very quickly and to derive many mags out of metagenomes, also methodologic approaches should be reasonably fast. In a sketch, this problem can be solved as a typical machine learning problem. So that's not very specific at this point. So the input data are metagenomic bins. We need a machine learning approach that can be trained by expert knowledge. This knowledge, it's of course one of the limiting factors here. This knowledge typically comes from literature. Sometimes it comes from databases or from high throughput experiments, or there are phenotype arrays, for instance, for certain traits available. But most of the interesting information for training comes from literature. And as usual, the negative information or the false information here is even harder to get. So you find, of course, a lot of literature about ammonia oxidizers, but to get reliable information that a bacterium living in the same environment or maybe being related is not oxidizing ammonia is hard to get. So the negative data here is harder to get. And so here, the expert knowledge actually from microbiologists, from biochemists actually in the fields were very important for us in establishing the first collection of models here. The outcome is actually a binary prediction. So far, we never worked actually on anything else than a binary prediction. Of course, we can think about that. So also turn that into quantitative predictions, rates, and so on. But I will show you that we're pretty limited even at that level. So the colors that you see in this table here, these colors, these colors correspond to the colors of the genome bins. So each genome bin gets just a row here, and per column we can then just make a binary prediction that a trait is present or not. And already as a spoiler, very often we have to say we don't know. So there's a third option, of course, to not make the prediction. We started and we published the first paper actually in 2015. We started with an existing algorithm that was designed for complete genomes, and we adapted this algorithm and we expanded very much actually on the training data. That was quite successful. The algorithm was Pika. Pika uses protein family memberships as features. So for all who are into machine learning, that's kind of how we make the features. So we just take a genome, we predict the protein sequences in this genome, and we assign these protein sequences to orthologous groups or protein families. Here you are, at least as long as it's about protein families, you're entirely flexible. So we can also incorporate, and we did it for a couple of specific predictions. You can incorporate also specific protein family databases, for instance, databases on marker genes for antimicrobial resistance. So here we are entirely flexible. The feature space is just the yes/no information that a particular protein family or orthologous group, homologous group, however you want to define it, is encoded in a genome or not. The traits, as mentioned, can be quite broad, can comprise roles, phenotypes, resistances, and so on. As said, that worked quite well. So we could kind of extend mainly towards incomplete genomes. We could really show that incomplete genomes can be handled here quite well. And we could also show that many traits can be predicted by that. I would like to skip all technical details here. As said, my interest is mainly to get the methods working, but of course, if you have questions, then I'm happy to discuss details here. In principle, it's not very difficult, the machine learning part. So we have used SVMs, we have used neural networks, we have used some other models. So as soon as we have feature-arrayed genomes, this is not a very, very difficult problem. The training, as said, was difficult, and so we spent a lot of time actually to collect the training data for models, and we put them into a database which is called FANDB. And what you see here, let's stay a minute actually on this slide, is that most of these traits are metabolic traits. That's due to the training data availability. So for metabolic traits, we have, as I said, we have these high-throughput arrays, phenotype arrays sometimes, that we can take learning data from, and we have a lot of publications. It's relatively easy to study molecular assays for metabolic trait, easier to establish than others. But, and that was important, in particular in the research of my group, we also wanted to go beyond that. So we for instance, included four, in my eyes, very interesting traits for symbiotic bacteria, which is obligate intracellular lifestyle, a very, very interesting trait to predict. This trait doesn't really work very well on incomplete MAGs, but as long as the MAGs are quite complete, we can kind of predict based on the genome reduction. So that's how these methods work actually. So you categorize the protein families, and as soon as certain typically housekeeping metabolic pathways are incomplete or missing because this is a microbe that has an obligate relation to a host, and it doesn't need to produce its own amino acids anymore. It doesn't need to produce its own nucleotides anymore. This is what you can see. And interestingly, we were doing pretty well with this method. So we could actually even predict other classes, other types of obligate intracellular bacteria than in the training set. So this was really, the training was really on genome reduction rather than on certain markers. So this was very inspiring for us. And we also solved this type 3, type 4, type 6 secretion system problem. So we could make reasonable predictions actually whether a functional secretion system of this type is encoded in the genome or not. There's only one antimicrobial resistance. Due to all the problems I think that you know better than me, very often it's not only the proteins, but it's also certain mutations and so on. So we just focused here on penicillin resistance. That worked quite well, but this is probably also an easier AMR trait. Some lifestyle traits, autotrophy, phototrophy, habitat compatibility was very interesting for us to study, like halophily, psychrophily, thermophily, and so on. And some physiology traits like motility, spore formation, gram-negativity, and so on. Some of these are very easy, like gram-negativity is super easy, is one of the easiest traits to predict. Others are very hard, like halophily or psychrophily. And in the last years, we could even utilize and further develop these models for applications, like in a joint project with the European Space Agency and with the NASA about planetary protection. Back to the bioinformatics. The prediction, as I said, is in principle a machine learning problem that you can even solve with different techniques that are available in machine learning. So this is not very complicated. Nevertheless, to really use that method, we needed to solve the feature generation in a reasonable way, and that was more a bioinformatic problem. So the baseline actually for feature generation, for annotating a bacterial genome and assigning the proteins to protein families is already needed for the first step in this prediction pipeline, which is the quality assessment. When we get a metagenomic bin for the prediction, we first need to know how complete and how contaminated it is, because the quality of the input data determines what we can predict. A totally incomplete genome cannot be used for anything. A very complete MAG can even be used, as I said, and gave you the example, can even be used for the prediction of obligate intracellular symbionts. And already here, so these completeness and contamination percentages required using the baseline methods, check M per bin, 10 minutes or something like that, with quite some memory. And furthermore, or further down, more of these annotations were needed for the prediction of the traits. So this was not acceptable. So we first played around in the initial years with alignment-based methods. So, for instance, going from Cognitor, the very kind of baseline methods, to HMMs. The hidden Markov models were better in terms of time, or faster actually in the annotation, doing reasonably well, but still per genome, just an E. coli example here, just in the range of an hour or so, or even longer. This could be improved by clusterizing these alignment-based methods. This, from time to time, we still do it like that. So we just distribute hidden Markov model databases over different compute nodes, load them into memory, and then run the annotation in parallel. But even that left us with a couple of minutes for an E. coli genome. So that was still very long. And if you want to expand these methods to hundreds of thousands or even millions of bacterial genomes, then a minute per genome is quite long. And so we kind of branched out. As very often, actually, in bioinformatics, choosing the kind of primary problem that you're working on requires you actually to work on secondary problems. And also for us, so here we set out and spent some time to develop a framework for the much faster alignment-free classification of protein families. We called it deep NOC, so it's based on deep learning, so kind of embedding of protein sequences. As I think that's now pretty common in bioinformatics, nothing very, very special anymore. In the end, kind of to predict then after a couple of steps in the neural networks, the protein families. Here we call them COGs for the clusters of autologous groups, but you can, as said, based on the training data, you can replace them by any other concept. We mainly worked with the ECCB database, actually, as the sequence and family databases based beyond. ECCB 5, ECCB 6 works quite well for deep NOC. Yeah, we expected, of course, that there would be a difference in the prediction accuracy for small and large protein families. That was to be expected. That's kind of obvious in such a alignment-free approach, and that's what we observed also. So COG 500, COG 100 were just test sets based on different minimal numbers of protein per family. COG 500 is a subset, actually, of ECCB that only consists of proteins that have 500 members or more. COG 100 is a database with more families that have at least 100 protein members or more. And so what you see here is that the alignment-based methods, they're pretty stable. So for them, it's not so much a difference if it's a small protein family or a large one, as expected. But for the alignment-free methods, and also some kind of heuristics or very aggressive heuristics as Diamond, for instance, also showed some, Diamond is also doing quite well. Only here, it shows a slight decay, but the rest are all the alignment-free methods. So it's a clear separation first between the alignment-based and alignment-free methods, and we see a slight difference actually between different alignment-based methods. Nevertheless, it worked reasonably well because the y-axis here shows you accuracy, precision, recall measures from 100% to 80%. So please don't be upset too much. So it still works quite well, and this effect is kind of to be expected. We came up with a threshold also to minimize the number of false positives. These predictions tend to produce false positives if a new protein family is in a sample and doesn't fit to any of the trained protein families. Then it tends to be assigned to some of the existing protein families and creates false positives that we don't want. So here, kind of with a prediction confidence value internally, we could come up with a reasonable cutoff that prevents too many of these false positives, of these wrong affiliations that we would like to avoid. Interestingly, when comparing the performance of the trait prediction back to the primary problem for alignment-based created features and alignment-free created features, there was almost no difference. That's very interesting for us. It was very good, but I think it's also an important learning that if we don't focus actually on perfection in the feature generation, we know that some of the protein families that might be important for a trait might not be predicted well because they're too small. They might not be even predicted at all. As long as there are sufficient other features available actually for the prediction of our primary problem, then we can live with that. And that was very, very helpful for us. Although we know the limitations of DeepNock, if we would take DeepNock as an endpoint, if you would, for instance, do a functional genome annotation based on these alignment-free protein family associations, I would be very skeptical. But we don't use it as an endpoint. We just use it as the feature for the feature vectors actually of our genomes, and that's good enough. And that's and it's much faster. It does the prediction in seconds or less than a second actually per genome, compared to a couple of minutes per genome before the alignment-based methods. And so here, I would really argue that even imperfect kind of methods for the feature generation, as long as there are many other features that can compensate for that, can be very useful for problems such as the trait prediction. To come to an end here, the more than 50 models that I showed you for different traits and roles can be predicted very reasonably in metagenomic beans, either based on alignment-based features or alignment-free features, DeepNock. They are always presented to the user as a table, as I showed you, so a table of kind of the traits versus genomes, or in this case here, it's a more detailed trait prediction table where the prediction for one genome is shown yes, no, with a certain probability and a balanced accuracy that we know can be achieved for a certain completeness and contamination. That's a bit hard to understand, so I would like to come back to that in an example just a minute later. The software, one software tool I mentioned already, which is crucial, which is DeepNock. I can really recommend it. And the other part of the software is PhenoTrax. PhenoTrax is just the machine learning part of it. And DeepNock and PhenoTrax together allow you to do the trait prediction. There was, for 10 years, a web interface, which is fendb.org. At the moment, we had to switch it off because it was already 10 years old. And we had to switch it off because the platform that we used for this website was not sufficiently safe anymore. At the moment, we are kind of re-implementing this. On the website, we provided a lot of query functions to the data that were used for training and also used or generated by prediction. That's, for me, very important. Now we call it explainable AI. You all know that. But when we started with that in 2015, it was already clear that it would be very interesting to provide queries by trait. For instance, for biochemists. Biochemists who would like to understand a certain metabolism, or molecular biologists who would like to understand type 3 secretion systems. It's important for them to see what are the main features that are responsible for good prediction based on presence and absence, which means we can discover, for instance, new potential regulators or some other accessory genes around them. So we provided on this website these kind of lists by trait. This was very, very interesting. Same we can do by feature. So we can just check one of the protein families that typically occurs in bacteria and can ask, okay, in what traits is, or for what traits, which traits is this family more or less important? So we can rank kind of the traits for a given protein family. That's, for instance, interesting for protein annotation. If you get across a certain, maybe not well annotated protein family here, you can get a completely annotation independent statement in which traits this protein family might be involved. And of course, we can also go the kind of usual way by genome. So we can ask, okay, this is a given genome. What are the trait predictions for this genome and why? Right? We always shared all training data. Nowadays, this is not the only trait prediction approach anymore, but I think there's not much training data around because it's so hard to get from all the literature studies. But for FenDB, as long as the website is now under construction, I'm happy to share all the data with you directly. So that's, we hope to get by the end of the year with the new web portal for that. Last but not least, let me share with you an application. The first larger scale application that we did was a microbiome study in which 150,000 MAGs were reported. It was a meta study. Many of you will probably know it. We chose this study just as a source for a relatively large number, at least when it was published, it was a very large number of metagenomic bins that we can start to study. When we featureized them, we used DeepNock for that, as I explained, and then run all the more than 50 FenDB models over all the traits. Then we get tables like this. Let me explain a little bit what these tables mean. First, of course, we get an organization by a particular microbiome sample. So these are the background kind of colors here. So this is the end of one sample. I just selected this for the screenshot here because it's here's where one sample ends. Then there's another sample, and here's another sample. So each sample comes with metadata like geographic information, ethnic information, some health information linked to it. So this is the kind of sample. The samples were very unequally represented. So here, this is a relatively small sample with only a few genomes being reconstructed out of it. And per genome bin, you see a row here. Each row is one of the genomes that is present in these samples. In all these columns, you see then per trait or per role, one of the FenDB predictions. And interestingly, most of the dots are not colored, which means no prediction could be made for this trait because the genome bin was too much contaminated or was too incomplete. For us, very important to have this option to say, we don't know, right? So wherever you see gray, then no prediction could be made. Like here, for instance, these two genomes that I show you, they were obviously quite not so high quality, and so only a few predictions could be asked for. The rest is then kind of green, red. So yes, no, just a binary classification. In total, yeah, using some thresholds for completeness, we did predictions on more than 100,000 MAGs. This is just the distribution of yes, no, and we don't know across all MAGs and all traits. Some of these traits were kind of used also as kind of internal controls, like anaerobic lifestyle, for instance, is to be expected in a human microbiome. And so this should always be one. Somewhere it is, somewhere it is and it worked out. I think it's here. It's this one where we just see a more or less green bar. So some of these traits are kind of obvious and can serve as kind of internal controls, or at least where we have expectations for. Others were open. There are many ways to analyze these data, and here I think it's now we now get into the area where also CAMDA is working on. So how to assess methods and how to work with them. This is a t-SNE plot of all the genomes by their trait similarity. So each dot is a genome, and of course there are big clumps here. That's clear. Each genome is represented of the vector of the trait predictions. Typically, you see these features per sample and per. Sorry, that's per sample, not per genome. Sample and the affiliation. That's a sample plot. You see typically sample plots, and then the feature is by taxonomy, for instance. Here we have the trait predictions. And so there were some interesting areas that we inspected closer, where we found, for instance, the infant microbiomes. There were not so many samples actually in this data set, and we did some other studies actually for disease. Same. Just to give you one example here, what we found for the infants, so for the very young microbiomes, that was an increase actually in the type 6 secretion system roles. This is not very obvious when you look at the taxonomy, but when you look at these roles, then the enrichment actually in type 6 secretion was very clear. And for microbiologists, that also makes sense because type 6 secretion is a secretion system that not only is involved in the interaction between host and symbionts, but also is a defense system between different bacteria. So it makes sense that we see initially, when a microbiome is growing up in an infant, that we see an increased kind of competition between bacteria where the immune system of the host is not so much involved, like later in us here, mainly, or the immune system of the host is controlling a lot of the microbial composition, but initially probably also interbacterial competition is stronger. And so we had a much higher proportion of genomes with type 6 secretion systems initially in these very early microbiomes. And this leaves me actually now with kind of some summaries, but also some questions actually that we don't know, at least not very well yet. So to summarize, I can say that even complex traits can be reasonably and quickly predicted from metagenomic bins and mags nowadays. So this works reasonably well. FNDB models can be very well explainable. It's very interesting material basically to work with and to try to understand the roles of genes, gene families, and genomes. So for us, this is one of the most interesting aspects. Of course, FNDB cannot predict completely new mechanisms as usual. It's a machine learning approach. We have learned this lesson in our institute quite well because I showed you the example of ammonia oxidation, and some of you might know about COMAMOX, a very interesting trait that was discovered recently, and we could have predicted COMAMOX earlier because we were working actually on a couple of these microbes, but we couldn't see it. So COMAMOX for us was always kind of classified as ANAMOX. So we have learned this, or it's to be expected anyway, but we have actually really made this experience. At the moment, just a disclaimer, it's only available for bacteria just based on the limited genome and maybe mainly also limited training data. And for archaea, if you look at archaea, then typically actually the traits are very much linked to taxonomy. So it's probably easier to just do a taxonomic classification and then to work with that. And the question, as said at the moment, that we cannot really answer is a quantitative comparison. It's already hard to compare microbiome by the taxonomic composition. But here to make a comparison between microbiome based on trait predictions, how to incorporate the abundance of the microbes in a reasonable way, because we don't know the actual activity of the microbes. So this is, for us, a very open question and something to work on in the future. Implications for CAMDA, just my thoughts. The microbial genome drafts are certainly massive data, that's clear. And we know that we can also reconstruct them quite reasonably. I mean, a new critical assessment of metagenome interpretation might be interesting, but in principle, we are now going for long reads, and so the kind of problem of data kind of quality gets at least partially less in the future and smaller in the future. The trait prediction is an interesting aspect also for an assessment because it's in principle open to many different tools and techniques. I have just presented FNDB here. There are some others, like the DSMZ also provides machine learning for that. But of course, you could also use metabolic modeling for predicting some of these traits. So in principle, it's an interesting field where multiple different methods, also different bioinformatic kind of approaches could compete to the same end. So that's what I also like very much. Even manual curation of genomes, like some microbiologists do, could be part of this competition. Unfortunately, the traits themselves are not massive data yet. So we have the typical kind of very asymmetric situation between the genome data and the functional data. The functional data can come from different traits, for different traits from different methods like cultivation, some biochemical assays, some high throughput phenotyping in some metabolic traits or AMR. So obviously at the moment, and that's my last point for CAMDA, it's probably most feasible, most reasonable to work on selected traits like you already do, AMR, but maybe also on some other traits in the future. Or that would be the alternative to go for the kind of for a description of the entire metagenome, like kind of health features of the host, actually. So where we don't take into account a particular trait, but more assess kind of how trait prediction would compete with, for instance, taxonomic profiling. So with that, I'm happy to discuss at least some of these points in your questions, and I'm open for discussion now.
Thank you so much for a very nice presentation and just showing the whole path which we need to go. So now we have time for questions. There are microphones on both sides.
Thank you for this very nice work. I have a quick comment about the climate challenge. If we compare with CASP, they provide a new protein that is not in the database for the tools. So that avoids for the tools to be overfitted for that new protein. But in the climate challenge, you use the same database that are already available for each tool, and you predict the simulated samples from that database to evaluate the tools. So do you think that we could find maybe another solution to better evaluate the tools?
May I ask back, what part of the CAMI challenge do you mean? Actually, you referred to CAMI 1, and you argue that the test data is based on the same database than the training data. Is that, did I get this?
For the taxonomic challenge, for the taxonomical part, when you just quantify the which cases is present.
No, actually. So there was for CAMI 1, we spent quite some time actually to get samples actually that were not published. So this was exactly following the same idea like CASP. So we had, and that was kind of maybe a limitation, we had from different sectors, from different institutions who worked on different kind of groups of microbes. We have had unpublished data for these genomes that were not published, that were not in any database. We generated reads using kind of computational sequencing. So we simulated reads, but still these reads were from genomes that were not published. And then they were open to everything. They were open to ISMB, binning, taxonomic classification. But I would not say that this was an easy task, because there were at least some members. I know, okay, these test data were, or competition data were biased, but there were at least some species included that were quite far from anything that was described before. But it's hard to get these kind of data, and that's also what limits us. But I think for at least for CAMI 1, that worked quite well. Because I fully agree, we need kind of unpublished data, and we need data that is sufficiently different from existing published data for a reasonable assessment. And that's what we did for CAMI 1. But this was also limiting then for further CAMI challenges, because every time to get these kind of data is difficult.
Okay. Other questions?
So, thanks a lot for the nice overview. I have a question. So, in the last 2 days, we have seen a lot of AI agents for a variety of different applications. So, I was just wondering, like, how far are we for different, like, building AI agents for there's a multiple microbial traits prediction or something like this, because I think which will be quite useful in clinical settings like this.
Yeah, thanks for this question. I mean, yeah, you need to train these AI methods, and here the training data, as said, traits are not massive data yet, and it's hard to get them. I think the most interesting data here that you can really do a lot on is available to companies. I'm aware of a couple of data sets worldwide that have been collected in different clinics and have been typed for antimicrobial resistance. And these data sets are, of course, very, very interesting, but these are not open source. They are not available. I hope that this will change also over the time, but at the moment, I think it's in a couple of pipelines in companies. Some of my students are working in these companies, so I know that. So we will see predictions, probably not based on that methodology that I showed you, because if I'm only up to AMR prediction, then of course I can also more streamline kind of the approach. Here we needed to be very open to very different traits, and but I cannot solve, and I can also not provide any solution in the future for the lack of training data. So this is more a political question. That's data that are collected, for instance, in public hospitals, and are then typed, are then also quickly getting available. So that's kind of the same like for the genome data, where we now have clear standards when a genome should be disclosed after sequencing. And I hope that the same will happen also for the traits.
Any further questions?
Yes. So you said with the example of ammonia oxidized bacteria, that's very easy because it only depends on a single marker gene, correct? But when in your actual model, your accuracy was not perfect. So I'm just wondering, would a marker gene have done better than the model? Did you look at the cases where your model predicted ammonia oxidation and it wasn't correct?
Yeah, just to repeat, I hope I understood it correctly. So it was about the marker gene-based traits, for instance, ammonia oxidation, and how they kind of work out in this concept. It works kind of, or it reflects each other very well. So if there is a particular marker, like the ammonia monoxygenase, the AMO gene for the ammonia oxidation, then it's typically also the feature with the highest weight in a model. In FNDB. So we have, of course, checked these cases, and typically it worked very well. But you see many others as well, potential regulators, etc., genes, whatever.
Well, specifically asking why in that case it's not perfect. So you gave some accuracy results. It wasn't 100% for ammonia oxidation. It just seemed interesting.
We have determined kind of these accuracies by randomly subsample genomes. And of course, there's a chance that we, of course, also remove the marker gene. Maybe that doesn't happen so often, actually, in natural microbiomes, but for this assessment, basically here, we just downsample genomes and work with 50% of a genome, 70% of the genome, 90%, and so on, which means there's a chance that you not only miss the marker, but you also miss kind of many other genes, and then you can make a wrong prediction. But in principle, actually, for these marker-based traits, when you really look at predictions and work with them, typically they agree quite well. So here, what you see is typically kind of the more the incompleteness of the genomes. That's the problem. Then actually, kind of that the marker gene is not visible. Yeah.
Thank you for the work of your group and yours. I wanted to ask about some history stuff, which means in old times, 20 years ago, people used to, when something was missing, as you mentioned, that some traits cannot be, the new ones cannot be traced. But people were searching for the traits that were known from phenotypes, then searching the genes that were missing function and then trying to match one to another. I wonder if this kind of old school methods can can work in these, using the methods.
Thanks very much for this idea. I fully agree. And it was always, yeah, I was always a fan of kind of using our predictions also to kind of prioritize targets, for instance, for experimental characterization. I've seen that a lot for bacterial lineages. That's what I think what reflects this already, so that certain bacterial lineages have been prioritized based on their metagenomic abundances, but lack of knowledge actually for characterization. But to be honest, it didn't work out that well. There would be a very obvious application of the trait prediction, and that would be the genome-based cultivation. I didn't touch on that just for time reasons, but in principle, if you say, yeah, there's a whole group of species, we don't know what they do, but we can at least predict some of the roles. We can at least maybe predict somehow how they could be cultivated. Then we could help each other. So that's what I would really like. I think the trait prediction alone will not solve the genome-based cultivation problem. We also need metabolic modeling for that. But I think here there's a big potential for bioinformatics to come up with good and well applicable methods for genome-guided cultivation. I mean, it's not a new idea, obviously, but I think here there would be the highest gain for the entire field. Because once we are able to cultivate, then we are much better able to study. Also, for instance, taking transcriptomes and so on. And so I would like to work into that direction, to be honest.
Okay, so thank you so much for the time.
Okay.